Yes, changing color is at home.
Okay. So, sorry to disrupt in the middle, but we can probably continue while you're reading. So it's supposed to be an informal talk, so feel free to ask questions in the middle anytime if you're confused or not following up at some point. Okay. So I'm going to talk about catching stem and progenitor cells using Boolean logic. I'm sure people here are familiar with Boolean logic, but most people may not be familiar with stem and progenitor cells. I'm going to describe what it means in easy terms. And I'll briefly talk about my background, so this is going to the outline. I'll talk about my background first and then go over all the rest of the talk. So my background is from computer science, and specifically in this case I'll talk about the Boolean analysis part, which is relevant for computer science. I was also in the electrical engineering department here, working with the computer science faculty. So in the electrical engineering world, we had finite state machine models for Boolean circuits. And there is something from there I can bring into this talk. I also developed some statistics along the way. And we'll talk about that. And so people can hear me fine? Yep. Or should I be allowed more? Okay. And so the main application is in the stem cell biology. And we'll talk about the details of the stem cell biology. So in the field of computer science and electrical engineering, I was working on this area of formal verification. What it means is a model checking digital system. So model check is you have a model and you have a property, you check, and there is algorithms. You develop new algorithms to check digital systems. And I had a lot of training and expertise in this area. I developed many different kinds of efficient algorithms to do model checking digital circuits. Then I developed these statistics to enable some of the analysis that I can do here to bring into the field of biology and medicine. And I along this way, I met a very talented postdoc from the Weissman lab here at Stanford, Siebel. He is actually a cardiac surgeon, working in the field of stem cell biology. And I started talking to him. I was very excited, and I realized that I should focus on solving some of the problems in the area of stem cell biology using some of my own expertise. And there was a connection there, so that basically bootstrap this whole process that I'm going to talk about. So the simple thing here is Boolean analysis. I think everyone here is familiar with this, but just for completeness, I'll talk about it. It's a mathematics of two values, and we have simple operations, Boolean operations, and or not. I will mostly focus on this type of operations called Boolean implication. What this means is x implies y, and in order for this statement to hold, if x is true, then y has to be true. Right? It's very simple for you guys. This is an example of a large digital system. This is what I used to work with. As Elliot mentioned, the error rate here is 1 for a billion, and similar to what we encounter in biology in DNA replication. So here I was working in this model checking digital system. In its very simple terms, what you have is this circuit, right? So you have this circuit, and there are these switches that turn on and off, and it changes the state of the circuit. So there are many different states, and changes the state when this is turned on and off. In another simple terms, what we have is this model of the circuit. So this is finite state machine description of the digital circuit. And you have certain properties of the circuit that you want to prove on the system. So these are certain properties are very simple, called invariants. Invariant are properties that holds in every state of the system. And these are encoded in state variables here. So you have x, y, and z, and these are the values. And you can see that x implies y is an invariant in this model, in this particular circuit, right? Because when x is true, when x is 1, y is 1. Right? Wherever you see when x is 1, y is 1. So this formula holds true in every state of the system. Right? So that's a simple property that you can check in the system. Usually the problem is bigger than this, right? So you have state explosion. If you have n variables, you have 2 to the power n possible states. And you need to develop efficient algorithm to be able to check properties like this in every possible state of the system without explicitly enumerating the state. Because if you go through each state, it will take forever, right? You may not solve the problem until your lifetime if you try to go through each state, right? So we develop algorithms to do this, solve this problem without explicitly enumerating the states. They are called symbolic computation. Right? So you work with symbols and formulas. So this is the background of the whole thing, and the theory and the Boolean logic behind doing this kind of computation. I'm going to apply to understand stem cell biology.
Can I say something?
Sure.
Yeah. So I think this formal method stuff is really useful for finding bugs in microchips. So if you guys recall back from I think 1996 or so, Intel had this very, very famous bug where it was adding numbers wrong. So this is kind of one way to ensure that your microchip doesn't add numbers incorrectly.
Right. Yeah, there was a famous Intel bug, right? So people were motivated to do this kind of analysis, and there was lots of funding from big companies like IBM, Ritsu, Intel, do formal verification, do research in formal verification.
Just make sure that your bank account doesn't get drained.
Yeah.
Because of some microchip bug.
Right. Okay, so let's start with stem cell biology. So in very simple terms, what we have is cells, right? These are cells at different stages. So there is a cell called stem cell, and the stem cell has two very crucial properties, two important properties. One property is the self-renewal, which if it divides, then it becomes, one cell becomes its own type. So it renews itself. It's called self-renewal property. So the stem cell can live forever, right? So whenever it divides, one of the cells becomes its own type. So it never loses its property, right? So the stem cell never loses its property. It divides, and one cell becomes its own type. The other cell can change its property. It's called the process of differentiation, right? So it becomes different from your parent. And if the cell is different, it goes through a path of differentiation till the terminal state. At the end, it becomes a terminally differentiated cell that may or may not be able to divide at this point. And most of the cell in our body performs, they are differentiated cells, and they perform all the important functions in our body. So these are very important cells, but to replenish, or if you lose any of these cells, you need stem cells, because they cannot do it by themselves. They are differentiated, right? So you need stem cells to make the differentiated cell. For example, you have all the blood cells, B cell, T cell, hemoglobin, all these or the cells are made from hematopoietic stem cells. There is a stem cell called blood stem cell that generates all the different cell types of the blood. So with respect to genes, you're looking at some molecules here. So DNA, you're looking at genes in the DNA. And there are genes and there are RNAs. And these genes are copied through a process called transcription. And they make several copies and make protein at the end. Right? And the protein is what the cell uses to do all its stuff. So in this process of differentiation, you have certain genes called A, B, and C here that are expressed, and they are made in these cells, and they are not made in these cells. Right? And there are other genes that are made in these cells. So the proteins from these genes are made in these cells, and they are not made in these cells. So these are called gene expression. And you can see that these genes are mutually exclusive. So whenever one gene is on, we call it on and off in this case, other gene is off. Right? So there are some properties of this gene expression. And you can have another gene that can be on here and then stay on for the whole duration of this differentiation. And these properties are, you can describe in simple Boolean logic. So for example, you can say if A is high, right, if A is on, then F is off. Right? So there's a very simple Boolean pattern in this process of differentiation. So I'm going to use some of this principle and then discover new markers of these stem cells and progenitor cells, and these are called progenitor cells, and the differentiated cells, using large data sets. Okay? So we need to apply some statistics. So first, you need to convert the values into binary form. Right? So here you will have data. So we have gene expression data from 60,000 samples. Okay, so these are all kinds of samples. There are normal, cancer. Imagine that you're looking at the entire universe in here. Okay? So we have samples from nose, eyes, blood, leukemia, colon cancer, breast cancer, ovarian cancer, all kinds of cancer, prostate cancer, and normal samples, cell lines, everything that people have done experiment on. Right? So imagine you're looking at the entire universe there. And we're trying to come up with Boolean states of these genes. So for CTH1, the values ranges from 4 to 14. And one way to do this is to sort the values from low to high. And what I do after this is to fit a step function. So if you fit a step function, you can draw a threshold around this line. And around this threshold, you put some intermediate region, and you call it low and high afterwards. It's a very simple step to binarize the gene expression values. It sort of works at this point, so we didn't complicate it further. Right?
So what happens when you get in the intermediate? Is that sure?
So all the intermediate values are ignored. So they are ignored, they're deleted from this data, right, for the analysis work. So after this step, we look at a pair of genes. So let's look at two genes here, ACTP and CABRB1. And here we are looking at the entire universe again. So we are looking at every single possible data out there. These are all gene expression data. Every point is it's coming from a microarray experiment that generates this kind of data. And these are all kinds of samples. And you can see that there is an interesting pattern in here. Right? There is an interesting pattern, and these patterns you would like to capture in a simple description. Like maybe the simplest description that you can find for this kind of pattern. So you can go through the ideas of Boolean logic. So here you can think about this pattern as saying if ACTP is high, then GABRB1 is low. Right? So that's a very simple description of what you can see here in this catalog. And we call this Boolean implication relationships in this case. There is a statistic developed to capture this kind of Boolean relationships in the data. Right? So if you go through this data set, there are many different kinds of patterns that you notice, right? In terms of,
So you segregate it statistically using the median or the mean?
the step minor effort, the one that you
Yeah.
here. So you have high and low, right?
But how do you determine the threshold?
You fit a step function. You find the position where it fits the best, the step. So go through each position, find a step that fits the best based on an error method, right? So sum of square error is what we consider here.
But what if you have a curve that doesn't fit well into a step?
Then it will just fit in the middle, right? So let's say if you have uniform values from 4 to 14, like say straight line, right? If you sort them, if you fit a step function, it will fit exactly in the middle,
Okay.
right?
Okay.
So it's not mean or median, right? So if you consider mean here, right? And if you have a lot of low values, the mean would be shifted down, right? Because there's a lot of low values. If you consider any other median, then it might be the low one, right? Somewhere in the low. Any other simple method like mean, median, mode is probably not good enough for this kind of thing because we don't know the distribution of these high and low. Right? And this is probably the best. Simple. So after these thresholds, that the lines that you see here are coming from those step lines. So you have 4 different kind of asymmetric patterns and then 2 symmetric patterns when you look at this kind of data. So in these data set, you're looking for these quadrants that are sparse. Right? So if you have one sparse quadrant, you call it a Boolean implication. You can have 2 sparse quadrants. You cannot have 3 sparse quadrants. Just the way I define high and lows. Right? So you need to have high and lows. Whenever you're looking at 2 genes, so you can have only 1 or 2 sparse quadrants. It turns out most people, when they do their analysis right now, they focus on these kind of relations because they do correlation. Right? And correlation will miss this kind of relationship. And these are asymmetric, right? So there is an inherent asymmetry in this picture. And most of the interesting biology are actually captured in this kind of asymmetry. And I'm going to ignore all of these kind of relationships and focus on only these kinds, which people have ignored for a long time, right? And identify these markers of stem and progenitor cells.
How are you picking the pairs? Like,
It's
what's the
simple.
relationship?
You can complicate further. You can have 3 and you can have 4, but
Is there a relationship between like the ones that you showed together?
yeah, these are pairwise relationships.
So
Right.
like in terms of the gene, like what are kind of the general functions of?
What is the general function of this gene?
Yeah, like the specific pairs that you choose to do. Like what's the
Oh, these pairs
relationship?
have a significant Boolean implication relationship because they have one sparse quadrant. Right?
Okay, but
So-
so you don't choose them based on related functions, or you?
No. It's completely from the data. The things that I ignored from here is the pair that don't have any relation. That means they are all over the place, right? So if they're uniformly distributed in all quadrants, then they are not fit.
How many different gene expressions were you looking at?
Sorry?
How many different gene expressions were you looking at?
The pairs, how many
Yes.
different pairs? 3 billion pairs.
Okay.
So out of 3 billion pairs, 300 millions have these kind of relations. Around 300 million. And most of them are not like related.
So actually, really quickly, just back to the step function thing.
Yes.
Have you ever seen patterns where there's more than one step, or 2 steps, I guess? If there are 3, like, or 4 or more?
Well, if you try to fit it, you will see, right? So
Well, still, but if there are 2 inflection points, then how would you decide which one?
at this point, I just look at whatever the step minority, right? So we are blind to that
Okay.
kind of pattern. So if it's actually fitting two different steps, then it probably fits one of the steps. Would be the best. Depending on the number of values in the middle, if the number of values in the middle is very high, then it will fit in the middle of that. If the number of values are small, then it will fit to one of them.
So basically, you would just ignore one of them.
Yeah.
Okay.
Yeah.
What's the rationale of only using pairwise relations?
It's just to make it simple.
So it's mainly because of computation limitations?
Computational limitation, because if you look at pairwise, there's 3 billion pairs. If you look at all triples, it's close to impossible, right? It's very hard to compute.
I'm envisioning that there could be some interesting.
There could be
higher
interesting
order.
ones, yes. There could be interesting higher order relations. That is also because of some of these lower order relationships that are combined together makes the higher order relationship, right? But there could be some intrinsic higher order relations which is not captured by any of these pairwise relations, which we are ignoring at this point. Okay. So now, if you think about this as an entire universe, right? Then these patterns are considered as invariant patterns. That, for example, for this case, you can say PTPRC is low, CD19 is low. Since you have looked at all of them, and only a few very rare cases, there might be noise, fall in this quadrant, you could imagine that there is nothing here, and say there is an invariant relationship between these two genes, and the relationship is if PTPRC is low, CD19 is low. Right? And we'll use this invariance to reconstruct the process of differentiation. Okay? So now we look at the stem cell biology. Right? So let's talk about differentiation in general. So you have a gene called A, which is expressed in the beginning of the differentiation, and a gene called B, which is expressed in the late stages of differentiation. And imagine that you have a Boolean implication between A and B. Right? So what that means is if A is high, then B is low. Which is certainly true in this particular path. Right? So this is a path in the system. Right? So if you think about the human being as an organism, right? There's several different processes of differentiation going on in our body, and it originates from stem cells, and there is a path of differentiation, and there's many different paths of differentiation. This is one possible path, and in this particular path, this property holds, right? But if you could somehow find out that there is an invariant property that could hold in every possible path of the system, no matter what is the path, right? Then you could use that as some priors to construct some of the new markers that I'm going to talk about in a specific example. So this is a path in our blood cell differentiation. This is called B cell. B cell fights infections and all kinds of foreign antigens in our body. It is specialized cell, differentiated cell, and starts from hematopoietic stem cell. As the stem cell differentiates in one of the paths, it becomes B cell. Right? So in the path of B cell differentiation, there is a property that people know right now. So this is coming from known biology. So there is a gene called KIT, which is expressed in the stem cell and the progenitor cell, and it goes down during differentiation. And there is a gene called CD19, it goes up during differentiation, and it is expressed in somewhat more B-cell committed cells. Right? If you look at the entire universe, so here you're looking at the entire universe, what you see is that there is a Boolean implication, right? You can see that this quadrant is sparse, that means if KIT is high, CD19 is low. Which is certainly true in this particular path. So you can imagine that this property might hold in every possible path of our system, right? So that's a powerful assumption, right? So if you assume that, you can try to predict gene X, right? So think about some unknown genes and think about a diagram that you want to have. So in this case, you can say that X is expressed or turned on before CD19 is on, right? So X is on before CD19 is on, stays on throughout. What you can do here is you can think about Boolean relationship between KIT and X and CD19 and X. So what is the Boolean relationship between CD19 and X? Let's see if people can answer this question. What is the Boolean logic between CD19 and X?
Whenever CD19 is on, gene X is on.
Yeah, it's very simple. So if CD19 is high, X is high. Remember, it's asymmetric, right? So it doesn't mean when X is high, CD19 is high, right? So there is an asymmetry in this picture. And we're going to capture the asymmetry of the gene expression to be able to discover such genes, right? So this asymmetry, and you can also have, if KIT is high, X is low, right? So by this logic, we basically say X has to be low here and X has to be high here, right? But X can be high or low in the middle, right? So this is a very simple logic, and you can do a database lookup, right? So by looking at all of these possibilities, all pairs, I have created a database of these kind of relationships. At this point, you can just do a database lookup to find what X satisfy these criteria.
So why don't you just also measure X at the same time?
You can measure X, but we don't have access to all of these samples. Imagine that we don't have access to all of these samples, right? We're not taking any measurement from any of these parts. We don't have any measurements, right? What we have is random collections of data sets, like completely random collection of data sets. It is cancer, normal, all kinds of garbage in there, right? But there is an invariant relationship in the data set. The invariant relationship is what we are trying to grab on. And we have this idea, we have this belief that there is something, there is a process of differentiation. And we're trying to characterize what happens in this path by looking at the invariant relationship. Right? So that's basically the idea of the whole talk, that you can use invariant relationships to come up with markers of differentiation even though you don't have access to these samples. In this case, we have access to all of these samples. So we can sort hematopoietic stem cells from our body. We can sort B cells from our, like purify the cells. But in many cases, we don't know how to sort cells from skin cells. We don't know how to sort the actual stem cells from the skin cells, because we don't know how the cell surface marker combination there. We know, it turns out the B cell differentiation is the only pathway where we know a lot about. And this is the best understood differentiation pathway that we have characterized so far. We have around 12 different states of differentiation that we can purify and measure. For most cases, we don't know. In the cancer, this is also, you can have a path of differentiation within cancer that you can deconvolute from this data that I'm going to talk about shortly. So in this case, you can talk about normal process of differentiation from this random collection of data. Okay? So it's very simple, it works. We have validated in many systems. In this case, you can also complicate it further. You can say combine multiple genes. In this case, you can say Boolean formulas defining particular state. Right? That brings to the question that you guys asked here. Can you come up with some higher order relations? Right? So this is looking at those higher order relations and testing specifically in this particular pathway, right? And predict some of these genes. So what happened was we went to the lab, we sorted the cells, and all the genes that we predicted, most of them were validated by this higher resolution qPCR experiment. Another validation was that there were actually, so we predicted around 62 genes in this particular pathway, and of these 62 genes, 41 of them have been knocked out in mice. That means there is a mouse without that gene. There is one gene deleted completely from that mouse. Right? It's amazing that we can have such mouse. And these mice knockouts are very useful for understanding the biology. Right? Of the 62 genes, 41 of them have been knocked out. So you might think that in some of these mice, people have looked at what happens in their B cell. Right? They have B cells in them. And if you look at literature, 26 out of 41 have specific defects. They have reported specific defects in B cells. So this is a confirmation that the gene that we predict purely computational, right? So we didn't use any direct sample from this particular pathway, but we predicted a bunch of genes from this random collection of data set using Boolean implications. And at the end, what we see that if you delete some of these genes, you'll have a defect in that specific pathway. So it's pretty important. Right? So the genes that you get are really, really important. And this is kind of gold standard for biologists to actually see that the genes that you get from a computational algorithm is of some use. And in this case, we discovered another marker and a new population of cells within the B cell differentiation. So we did a bunch of arrays in the blood cell. These are different blood cell types, and these are genes that I showed you. CD19 is expressed in the B cell image. So this is hematopoietic stem cell. And this is the B cell, this is the T cell, this is the NK cell. They are different blood cell types. And you can see that during the process of differentiation, CD19 comes on. This is how KIT is expressed. KIT is expressed in most of these cell types, but it goes down in this path of differentiation. And this one we have discovered a new population that actually turns on in this branch point of B cell and T cell. Okay. Let's talk about cancer. How do you apply this kind of concept in cancer?
Yeah. Question. So could you go back to the knockout page? So what will happen if you randomly knock out a gene from the mouse?
Randomly?
Yeah.
You won't see any phenotype.
Okay.
It's very hard for people to see a phenotype. People spend years, right, to generate the knockout. At the end, they realize that there is no phenotype. Right? This is what happens in real life, right?
Okay. So
that's why we burned a lot of dollars, like billions of dollars, in this experiment.
Yeah. Okay. All right. So that number is really significant.
This is very, very significant. This is really significant. It turns other genes that do not show defects in the mice, they have certain function in B cell, but there are redundancy in our system, right? So if you delete one gene, it doesn't really matter because there is another gene that takes control of the same thing. Right? Even with the redundancy, if you see such phenotype, that's really, really powerful.
Okay.
In C. elegans, if you delete a gene, you will see a phenotype very easily because there is very few redundancies. In mouse, it's very, very hard to find important genes like this. Okay, so how we can apply the same concept in cancer. So you can think about process of differentiation. This is an example from bladder cancer. So you can simply imagine that the cancer cell also differentiates, right? And there is a process of differentiation within cancer. It turns out here, in the bladder cancer, we knew certain things, certain markers before we did this experiment. We knew a marker called keratin 5, and there is a marker called keratin 20. And we knew that keratin 5 goes down during differentiation, and keratin 20 goes up during differentiation, right, within the cancer. So what happens is, if you take a cell that is keratin 5, put it in mice, it becomes a tumor in mice, and in that tumor you will find the keratin 20 positive cells. Right? So in this case, we wanted to zoom into more the stem cell area, right? The stem cell area is around here, so this is where the stem cell is. And we don't know what the actual marker of the stem cell is. So we want to be as close to the stem cell as possible, because that's where the most aggressiveness comes within the cancer. So you set up simple Boolean logic here. You can say you don't know this gene X, and you want to find a gene when X is high, keratin 5 is high, and if X is high, keratin 20 is low, right? In this entire universe. It's a very simple experiment, right? So you get a few genes because you're pretty stringent on these numbers. There's 60,000 values here, and this gene came to the top. Keratin 14 came to the top. So we hypothesized there are 3 different cell types in this cancer. So one could be keratin 14 positive, keratin 5 positive, but keratin 20 negative, right? Which could be upstream of this other gene, which is keratin 5 positive, and keratin 14 negative, and keratin 20 negative. At the end, you have keratin 20 positive cells. Right? So this is a hypothesis that you can derive from this Boolean logic. And we saw the same thing in the cancer. What we saw at the keratin 14 was a subset of cells, which is keratin 5. And we have sorted these cells through a bunch of cell surface markers. We can sort these cells, and we can grow tumors from the cells. And these cells grow tumor, and in the tumor you find these other cells. That means these cells became, during the process of differentiation, they became these cell types. Right? These cells also grew tumor, and they did not make these cells. So if you sort these cells, purify these cells, and put them in mice, these are from human cancer, human bladder cancer, and the mice grow the tumor. In the tumor, if you analyze the tumor, we call it xenograft. In the xenograft, you don't see the upstream cells. You always see the downstream cells. That means there is a process of differentiation within cancer. And it's very hard to come up with these kind of pictures purely from random guesses. And that's what people do right now. Right? And to get to this point, it takes around 6-7 years, more than 10 years in some cases, that you could sort some cells and identify the cell types and the lineage relationship between them. It's very important in cancer. And in this cancer, what we show that if you look at keratin 14, right? Keratin 14 has a very good antibody, and you can stain tissues sections. So when the patient goes to the clinic, they biopsy their tumors, and the tumors put on a slide, right? And there's a thin section of this tissue that you can stain for these kind of proteins, this keratin 14. So if it's brown, then it's positive. If it's not brown, then it's negative, right? So this is a very simple staining procedures that any hospital can do anywhere in the world. Every hospital has expertise to do this kind of analysis. So you could call a bladder cancer positive or negative based on keratin 14. And what we saw at Stanford, we had a collection of these tissues, around 150 of them. And we saw positive and negative, and there was a significant difference with respect to the overall survival of these patients. So whenever they are positive for keratin 14, remember the keratin 14 marks, it's close to the stem cell, right? Marks the most aggressive cells of the tissue. And when you see a cell type within a tissue, the patient dies quicker. Right? So you can almost predict the patient's survival from this kind of data. And it turns out at the Baylor, this is another hospital where we got bladder cancer sample. Traditionally, what people do is they look at stage and grade to treat patients. Okay? So stage is how far the tumor spread from the primary site. So it spread to distant metastasis, and stage 4, terminal stage patient. Stage 1, that's early stage. Grade is a crude measure of differentiation, just by looking at the tissue, how bad the tissue looks like. And stage and grade are the best marker that is used right now in the clinic. In the clinic, when the bladder cancer patient goes to the hospital, the doctor looks at stage and grade and gives all the treatment, makes all the treatment decisions. What happened in the Baylor data set, stage and grade were not predictive of overall survival, which is pretty amazing, right? So there is a hospital where people treat these patients and they are confused which patient is more aggressive, is bad, and which patient is good, right? In this hospital, keratin 14 was the strongest predictor of survival. And so here you can actually complement what people use right now in the clinic. And it turns out that every hospital can do this kind of analysis. So when we validated this idea on this marker in large cohort, like 5,000 patients, which we are trying right now, after the validation, everyone will start using it. Everyone will start using this kind of analysis to identify high-risk bladder cancer patients and make their decision on whether to remove bladder. It turns out that you can remove the bladder completely, and that's the best treatment for bladder cancer patients, if you can find out which patient has bad outcome, which we do right now using stage and grade. So this is a very general principle that we can apply in all kinds of cancers, right? So there's no, in the method, we haven't put bladder cancer in there, right? This is just a general platform that you can use Boolean implication to mine any kinds of cancer, which we are doing on all kinds of cancer. And there's an example from colon cancer here, which there's a lot of slides here. I'm going to just go through quickly, and you can just see there is a Boolean implication here that finds particular cell types and the process of differentiation, which we have validated in the actual normal tissues. In this case, we discover the markers using Boolean implications, and we go to the lab and then do this experiment and identify really important markers in cancer that we also validate using single cell analysis. And in this case, we also find markers of very important cells like bottom of the pit cells in this case. These are areas where the most aggressive cells reside in the colon pit. In most tissue, we don't know the area very well, but in this case, we know where it resides. So it's kind of easy to validate in this model. But you can use this same principle, same idea, using Boolean logic to deconvolute the cell type diversity and their lineage relationship between the process of differentiation, just by using simple properties.
How many types of cancer have you been looking at, and how many types do you find such a correlation in?
Every single one. Right? So you take any cancer, you can work on it. Right? So you come up with some ideas, start with some markers.
Well, like specifically for your data set, you showed 2 hospital grades for specific cancer where the results are really convincing.
Okay.
Like,
I'm going to
how many
show
examples of that do
another.
you have to look through before you got to that?
Oh, one. Just
So
one.
that was the first one that you
We
looked at?
tried the first one, it worked. Here we tried the first one, it worked. Right? So there is the process, when you look at this Boolean relationship, it reduces the noise dramatically. Because randomly you don't find this kind of relationship. And there is a reason why these relationships are there. And when you do experiment on the markers that follow these kind of implications, most of the time they work. And luckily they worked in the tissues when we look at it in a single cell assays, it works around 60-70% of the time, which is pretty remarkable. So in this case, giving you one example from colon cancer, where you identify another marker, CDX2, and you can see CDX2 is a marker of differentiation in this case, and CDX2 negative tumors have bad outcome. And it's also true in the multivariate analysis. That means it has some additional information to stage and grade. In this case, stage and grade are very powerful markers for identifying high risk, very powerful indicator of high risk colon cancer patient. And what happens in the key stage of patient is stage 2 colon cancer patient. They're considered early stage, somewhat more early. Stage 1 is early, but stage 2 is the next one. What happens in the stage 2 colon cancer patient, they will not give them chemotherapy. Right? So these patients will do well in future, so they won't give chemotherapy. But stage 3 patient, they all get chemotherapy. Right? Stage 3 patient, all of them get chemotherapy. But what happens if you look at the stage 2 patient, in the set of patients with CDX2 negative, they have bad outcome. Right? And in each stage 3, they all get chemotherapy. Right? So this CDX2 negative patient in stage 3 also have bad outcome. But when you give chemotherapy to these patients, they respond to chemotherapy. So what you can do here is you can extrapolate this idea. You can say, we know this patient, stage 2 patient, they are at the early stage of their tumorigenesis, and we wait right now. Right now, we don't give anything to them. And these guys most likely progress further to stage 3, and at that point they will get chemotherapy. And it might be at the worst stage at that point to give chemotherapy. Right? So if you can catch those guys early enough, if you can give them chemotherapy, they might do well in future. So this is another idea we're pursuing, and we have a clinical trial right now going on to show that CDX2 negative patient in stage 2 colon cancer is predictive of chemotherapy, and when you give them chemotherapy, they will respond, and some of them might get cured of colon cancer, which they might not get cured at this point. And these are other cancers that we are trying to, we're working on applying this kind of Boolean logic, and in each case, actually, I work with high school students who looked at some of these Boolean relationships and identified very important markers. High school student can do this kind of analysis. And it's pretty simple. It's really, really simple. And another high school students, this is 8th grade and 10th grade, and they actually were so excited that they developed Android application for this kind of analysis, like vector analysis.
Are these future Siebel Scholars?
These are the future Siebel Scholars. Yeah. So we have very simple ideas that you can actually teach high school students and get useful insights into cancer, stem cell biology, and some clinical biomarkers that you can use right now. So that's the whole idea. You can start with some high-level description of Boolean logic and go systematically to bench and the clinic. It's a very simple idea that you can use invariant relations. It's a simple tool to deconvert these tissue hierarchies and cell types. And along the way, you can identify very important markers that you can use in the clinic. You can use any kinds of tissues to identify these kind of markers. And not necessarily you can apply only on this kind of setting. You could also try to look at other data types. In this case, these are other kinds of sequencing data where you can use Boolean logic to understand the system better, some of the variations within the, in this case, looking at the exon expressions, and I'll give you some other examples. Here, HIV, you can also look at HIV, and some of you, I heard, works on HIV now. you can look at Boolean logic between variants of certain HIV, and there are actually Boolean logic rules between these positions. When this position in HIV becomes A, this position doesn't become C, right? It's something else. So there is a relationship between them. There is a reason why. And looking at this constraint property in all these viruses, you could get some insight into the evolution of the virus and potentially the targets that we can pursue for therapeutic applications. So that's it, and a lot of people to thank. I didn't do most of this work, but I developed the theory and the ideas, but these people helped validate some of these ideas, and without them it would not be possible. But I would like to thank Weissman, without whom this would not be possible, because he facilitated giving positions and support money to do the research. And also the Siebel Foundation helped me develop all of these ideas and move it forward to the next stage. So thank you very much for listening. And yes.
So, using your, method in actual, hospitals, not in experimental way, but like everywhere, what would be the time frame, from lab to the, everywhere, people are using?
Oh, for this one, that's a great question. So, if you have an immunohistochemistry, the staining that I showed, those are called immunohistochemistry. The staining-based assay is easily penetrating, right? So you can, because the expertise is available in every hospital, right? The idea is to validate on a large cohort, right? Here it's like only 200 patients or 300 patients. It's not good enough to trigger a large scale use, right? So you need to have a trial. First, a retrospective trial on thousands of patients, and then a prospective trial, right? The prospective trial is to make a decision on these using this staining, right? So can you make a decision that will improve patient outcome? And only after you show that there is an improvement in patient outcome using the variable, using the clinical indicator, as staining as a clinical indicator of high risk cancer patients, then everyone will start using it. So if you publish it usually we publish in New England Journal of Medicine, this kind of study, and if it's published in New England Journal of Medicine, the next day onwards, you will see it everywhere. You don't have to do anything. People will just do it. Because, yeah.
But would research hospitals pick up on this faster, like Stanford?
Yeah, because we developed it here, right? So. But people now are focusing on developing gene expression-based assays, which only Stanford Hospital or privileged hospital can do those kind of analysis. Immunohistochemistry-based assays is very easy and everyone can do that.
That's something really awesome about this.
Yeah.
The method is
you zoom into the most important guy, right? So that's the idea.
Right.
If we don't have a way to zoom into the most important guy, then we have a noise, right? We set up genes, and then hopefully if you combine all of them together, you get some idea. That's what the gene signatures do, right? And that's one way to do it. This is another.
So initially when you do the modeling for the step, how many data points do you need to get enough power to do that?
Well, in this way I use them all, right? So that's 60,000.
Like that's just what you curated from the databases, or?
Everything that's available in the public domain. Right?
What do you think is a minimum?
Minimum is around 100, 200. But
So-
the number is not enough, right? We need to have some diversity.
Right.
The diversity is the key. If you have only breast cancer data, let's say, right? If you have only breast cancer data, then you don't have enough diversity in the data set. So for some genes, it might be total noise, the threshold.
But you could still do the same analysis with a reduced set of genes, right?
Yeah.
Yeah. So I guess my point is if we use like the single cell technique, then we can do like 200 cells and then we can already do the analysis.
Single cell, and also there is some noise associated with it, right? So if the data is very noisy, right? So if you look at let's look here, let's look at one. So data is so noisy that so here I kind of idealized it, right? So in this case, I want to see something really ideal that are kind of invading. You may not be able to see that in the single cell data because it's so noisy. So even if there is a relationship like this, just because of noise, you basically have enough numbers here, right? And it's a different way of looking at it. You don't have a Boolean implication there. Most often you don't have Boolean implication because there is always noise in this data. There is a lot of noise in this data, right? Whenever there is noise, you will accumulate noise here, and you won't have an ideal situation like this.
But isn't that just a matter of setting the baseline differently?
No, it's not a matter. It's basically the noise level. In the single cell data, no matter where you set the threshold, and I don't know the technology at this point now, but I've looked at some of this data. No matter how you set the threshold, the noise basically is huge, and it doesn't make it ideal.
Maybe we can talk offline, or you can show me
Yeah.
what you've already done.
But you can relax it further and then relax it once. But then you're prone to more noise. Any other question?
Actually, with the application of this, like I can see, like you could simulate like how many kind of lives you could save. Like basically, when I work in tech and like we build products. And when we build products, for example, like I work on Google Maps, and when we implement this feature, we can save X% of those Y number of users, and that results in like some lifetime saved, right? And with this, like looking at the statistics of like expected lifetime, like the improvement you can make with this, I feel like you could estimate like with if we apply this to like X% of the world's hospitals, you could simulate like how many lives you can save, and that could be an amazing number.
Yes, yeah.
Have you ever tried that?
No, I haven't.
Yeah, that could be interesting.
Yeah, it's a theoretical exercise to see what's the
Yeah.
value of this.
Cool.
What's the value of this, right? So, so that's a very. Let me give you some insight into how to the business people. So that how can you make money out of this, right? So it's basically most of the time what happens if you look at a cancer, right? When you find a marker, most of the time you don't have a really good antibody against those markers. Right? So limited by availability of a really good antibody. Right? An antibody people make and it takes a long time. Right? And if you put effort, you can make it. Right? It's not impossible. Right? It's possible. It turns out if you don't have a good antibody, then it's useless. Right? So you cannot use the staining-based approach. You cannot use staining. But if you show a marker and you develop an antibody against those markers, then you own that. You basically own the patents for the antibody. Nobody else has that antibody. And when you show on a patient large-scale cohort on any of the cancer, everyone will want to use that antibody. So you sell your antibody to the whole world. And if you show value, if you show that you can save some life, it has billions of dollars.
So you use Boolean implication here, right? That's just kind of thinking about data mining and machine learning. There's also association rule mining, right? Or if you buy bread, you might also buy butter. Or if you buy flour, milk, and eggs, and vanilla, you might make a vanilla cake, right?
Yes.
And you thought about other kinds of mining other than Boolean implication?
Well, this is the simplest one that I can think about, right? And simpler you go, you can actually teach to high school students. If you try to go further, then you may not be able to teach. But on the other hand, what happens is talking to biologists is also very similar. It's talking to high school students. From my perspective, right? So if you talk to biologists and start spitting about computer programming like skills, C, C++, and things like that, they will never listen to you, right?
Building on that, I think there are a lot of applications in marketing because it's natural Boolean activity for does a marketing activity, does not, and does buy, does not buy, is the same as cancer, not cancer.
Yeah.
Yes, it's pretty
Multivariate
simple.
application.
Yeah, many other data sets.
Has there been any follow-up work? Has anyone cited the work and tried a different approach from what you've been doing?
No, actually, people work on their own approaches, right? And then they hit that so they hit the wall and then they skim around and see what other approaches they can find. So I've been trying to popularize this, right? And show that this approach, even though it's simple, it works, and people think it's too simplistic to actually apply and do something useful from this kind of analysis. So people have been ignoring this kind of approach, but now I've given so many talks around, so people are very excited and they are working in the similar areas now. So there will be in few years we'll have more similar kind of approaches. But right now people talk about this Bayesian analysis and other kinds of analysis, which is slightly more complex, right? So it captures, this is very simple. You can do the same thing in a complex framework, for sure, right? So Bayesian analysis or other
Causal networks.
Yeah, mutual informations and other things that people do, you can do the same thing. But the fact that it captures the complexity, you don't simplify the problem. You don't see how you can apply it to this kind of problem in this way. And also the idea of using invariants to come up with this marker is coming from this idea of formal verification, right? And nobody has this kind of insight. If you talk to the Bayesian people, they have different kinds of insight, like learning and different kinds of things from the data, but doesn't have this kind of thing, right? Because it's just a different paradigm, different ways of thinking, different ways of dealing with the data.
So in a sense, discretizing it simplifies the problem.
Yeah,
Have you ever found that discretizing it actually removes some information that was necessary to come
it
up with
removes
simplification?
a lot of information. So imagine, so I don't find all the markers, right? I find only few that are good, but it basically doesn't find everything that's good, right? So it misses a lot of things, right? Misses maybe 90% of the things, but finds 10% or maybe 5% or maybe 1%, right? We don't know how much the percentage is. But even 1%, if you show that it is good, it's worth pursuing in this case. But people are trying to capture this 90% of the information, and that's where it becomes unwieldy and more complex.
It's almost intractable sometimes.
Yeah.
It might even help you with the outcome to find these binary cases, like very obvious yes/no cases, because then it might make the clinical decision of yes or no.
It's simple.
Like easier
Yeah.
than more complex
Yes.
calculations.
Yeah. Yeah. It makes simple clinical decisions, and it's easy for designing clinical trials. If you introduce any complexity into the clinical trials, right? Almost impossible to recruit any kind of students or convince any doctors to pose any of the theoretical trials. You need to make it very simple. They just work on one variable, right? Plus and minus one variables, and then see what the equations.
So just curious, how many times have you, in the various systems you look at, have you found where a binary view is not the best view? So as you mentioned, what happens when you have a three-step data?
Right. Well, there's many cases, right? So I only focus on 5% of the things that I find through this microscope, right? But there's like all these 99% of them, right, have other kinds of relationships, more complex, and you can capture using more complex modeling techniques, maybe more states, right, or Bayesian, more complex, mutual information. There's other ways, linear correlation. There's also a recent approach of looking at some information theoretic approach to coming up with measuring redundancy in the data set. And it was in science to apply the gene expression data to come up with some pairwise relations. All these other cases, they capture more complex ideas, and if you think of something, you'll find it in the data, right? It's like a lot of data, and there's so much of things out there. If you think of any crazy ideas, you will find it, right? So it depends on where you start. Right?
How much of the discovery process here is automated? In other words, like which part, like, is there any part that human needs to be involved? If it's completely automated, I mean, like, finding what's the like good.
The staining, you cannot do using right?
Yeah,
so,
sure.
Yeah.
I thought like those are like...
These are data generated from the samples, right?
Yeah.
So you have a sample, and then somebody, a human being, you can actually automate that right now. Maybe extract RNA automatically from the tissues and the cells, and do RNA-seq, like single cell RNA-seq, what she does, is pretty much automated from the beginning to the end, right? Here, the part that is automated is basically this, looking at these rules, Boolean rules, and that's it. And you find a marker, and then you ask for help.
Yeah, just thought that, if it's almost, entirely automated, you can just do, massive computation, and then, you...
After the discovery, you can potentially automate. So if the staining thing, you can automate this thing. I mean, you need to get the staining done in a framework where the machine can take things, right? But after you process in certain ways, the machine can take over the whole thing. This was actually done using a machine, the staining here. But there is some manual process. Yeah. It's not entirely automated. You have to watch.
Yeah. Feeds exactly into my next question. What did the high school kids do?
They won several awards, like this Intel Science Fair award. So one of the high school students was.
I mean, do they just?
So Jackie is still in high school. Maybe now in 10th grade. She did this analysis when she was 7th to 8th grade. Ryan is in Harvard now, and he won Siemens, Intel, 2 of the Intel award, and he got into all the schools he wanted, like Stanford, Harvard, everywhere. And he highlighted this project, the pancreatic cancer project. Christopher will join Stanford this year, so he also won all the awards that Ryan won, all this Intel and Siemens awards. So they are going through this, and they will come here at some point, maybe.
Leads me into a question about next year's conference on education. How do we fix education?
Education, yeah. So you can get advice from your advisor. And these guys are really fantastic. They are really good at programming.
So they just did it by themselves? Or
They did it by themselves.
Really?
Yeah. I was like,
Oh, wow.
how can a 8th grade and 10th grade can do programming and generate this kind of thing, right? Which I would not be able to generate this kind of thing in my high school.
Were they taking a class at the time, or were they just...
No, they were taught by their parents from the beginning, and they were like programming from their at the age of 4 or 5, I don't know. Yeah.
Now, did they want to be computer scientists or biologists when they grew up?
I think, so Kritika Iyer is now probably in 10th grade, and she is very good at math, and she won some of the Olympiads, and she is also good at tennis, and she's a tennis champion in US. Some ranking, like national ranking. And Sonam is good in like some engineering, engineering. So they are both engineering focused.
Have you sequenced their genome yet? Or?
No. Well, that's the next. I think this is practice. You can turn anyone into these guys probably. Okay. Any other question?
I think the coffee's worn off, right?
Okay, thank you very much.
Thanks a lot.